Structure and function of biological macromolecules. So we're going to start off with protein structure, and that's basically just going to be turning amino acids into polypeptides and then into proteins. There are four main levels of protein structure, and that's going to be the primary, secondary, tertiary, and quaternary structures. All proteins go through the first three, however with the last one, which is going to be quaternary, it only happens with a protein that consists of two or more polypeptide chains. And just for a refresher, a polypeptide is an unprocessed or uncoiled or folded sequence of amino acids. So the first step is going to be primary structure, and this is determined by the sequence of amino acids which are held together by covalent bonding, or more specifically, the amino acid peptide bonds. And basically this dictates every other step that's going to happen. So if you look at this diagram right here, the amino acid sequence, that completely, if anything is out of place, that completely changes the structure and the function later on. So it's extremely important that they're lined up correctly. Next we have the secondary structure. And there are two main forms of this you will see. So the first one is going to be coils, or this is also known as alpha helixes. And it's just going to have the symbol of kind of like a little fish. So this is a delicate coil held together by hydrogen bonding between every fourth amino acid. So if you look at the diagram here, it's just a little curlicue, but it's going to have hydrogen bonds all throughout it to help out with its structure. Next we're going to have the pleated sheet, and this is also known as the beta sheet. It's just going to have a little bee with a tail as its main symbol. So pleated sheets are, they have two or more segments of the polypeptide chain lying side by side and are connected by hydrogen bonds as well between parts of the two parallel segments of the polypeptide backbone. So it's going to be it's kind of like a piece of crimped hair, and it's just going to have the hydrogen bonds all the way throughout it as well for structure. If you haven't noticed, hydrogen bonding is very, very important here. Next we're going to have amino acids tertiary structure, which is the overall shape of a polypeptide resulting from interactions between the side chains of the various acids. So there are three main structural bonding here. So the first one is going to be hydrophobic interactions. So basically, as folding occurs, nonpolar side chains end up in clusters, which you can see here. And don't forget that hydrophobic means that they're afraid of water. So they tend to be very out of contact with water. And in case you need help figuring out what kind of how to identify it, it's just going to be anything that has just carbons and just hydrogen. That is a very, very easy way to tell whether something is hydrophobic. Next, we're going to have van der Waals interactions, which help hold side chains together, as well as hydrogen bonds for polar and ionic bonds between positive and negatively charged chains to help stabilize overall structure. So it's going to be the same example as before, but it's also going to be this hydrogen bond right here, as well as right here with the ionic bond. And you can tell that's an ionic bond because it's connected through the charge right there. Finally, we have the disulfide bridges, which are connected, it's pretty simple, they're just connected by the sulfur right here. Finally, we have the quaternary structure, and this is going to be the overall protein structure that results from the aggregation of polypeptide subunits. So these subunits are just going to be what we just talked about. It's going to be alpha and beta. And this only occurs between proteins that consist of two or more polypeptide chains, like we talked about earlier. So an easy way to remember this is it's going to be a combination of pretty much everything. It's going to be a combination of the alpha and the beta just kind of mushed together. So something to keep in mind here is denaturation. So denaturation, it can be affected by the temperature, pH, or pretty much any other environmental aspect. Basically, the weak chemical bonds and interactions within a protein may be destroyed, causing the protein to unravel or lose its shape. This is not a good thing. but it is something that you should keep in the back of your mind. So next we're going to talk about carbohydrates. So the most important part here is going to be the alpha and beta glucose. So when you're looking at the linear structure in the center here, it doesn't look too interesting. There aren't many differences. But when you look at the two diagrams on the left and the right, their main differences are going to be between their hydroxyl groups. So for an alpha glucose, it's going to be on the bottom right, and for a beta glucose, it's going to be on the top right. And while this difference may seem like pretty much nothing, it's very, very important to, like I talked about earlier, the structure and function later on. This can completely change the way that it bonds to anything else and how it reacts later on. So next we're going to talk about structural isomers. So they differ in the covalent arrangements of their atoms. So when you look at it, they have the same amount of carbons and the same amount of hydrogens. It's just how that they're placed together that matters. And like I said before, the actual lineup of it is extremely important later on. It can completely change what it's used for. So it could be used for energy storage, or it could just be used for structure in anything. Or sorry, in a plant. Next we're going to talk about cis-tran isomers, which are also called geometric isomers. So atoms differ in their spatial arrangements due to the inflexibility of double bonds. So single bonds allow the atoms they join to rotate freely without changing the compound, but with double bonds, they don't allow this rotation. If they form, each carbon has two different atoms attached to it, and two different cis-trans isomers are possible. So if you see in the diagram right here, there are double bonds present, meaning that there are two different ways that this can turn out. So while these things right here are in different places, it can also be right here or right here, and that's the main identifying trait of a cis-trans isomer. And then last of all, we're going to talk about enantiomers. So these are isomers that are mirror images of each other, and they differ in shape due to the presence of an asymmetric carbon, which is one that is attached to 4 different atoms of or groups of atoms. An easy way to think about this is just that they're like the left and right hand of a structure. And then one important thing to remember is that only one is biologically active because only that form can bind to a specific molecule in an organism. Thank you so much, and I hope that this helped.